are in La Jolla, California, at the Science Network's offices, which is the former home of Francis Crick, and we're with a long-time colleague of Francis's, Sidney Brenner, who is in fact the 2002 Nobel Prize winner for Physiology and Medicine, but is also Senior Fellow at the Crick-Jacobs Center for Theoretical and Computational Biology at the Salk Institute, Senior Fellow at the Janelia Farm Research Campus of the Howard Hughes Medical Institute, and you're also the subject of this brand new biography by Errol Friedberg, and a whole bunch of other things which we'll get round to in a moment, so welcome. Does this bring back memories coming into this home?
Yes, I haven't been here since I think about a week before Francis died. Yeah. And so it does bring back memories, but it's very hard to reconstruct what it looked like then.
Yeah, I know.
So it's now very impersonal, I think.
Let's go back to the Nobel Prize, the 2002 Nobel Prize. There could have been any number of things for which you had received a Nobel Prize many years before that, which was 2002. As I look at the citation with Bob Horwitz and John Sulston, it says, for their discoveries concerning genetic regulation of organ development and programmed cell death, which seems strange almost, given your work on RNA, messenger RNA and so on C. elegans and so on. What's the backstory of this one?
I don't know, but there must have been some idea of giving it for programmed cell death, and then, but if you do the posterior recursion, then you have to give it to Sulston, who discovered natural cell death, which is of course what Bob Horwitz did, and of course you have to go one step back and give it to someone who started the means by which all the other things came about. So I think it was a construction, but as I said then, I thought it was to, as I said, there's a fourth prize winner here, but that is of course C. elegans itself, and I think the testimony for that is just what has happened to that entire field since it got started a long time ago, almost 50 years ago now, and I think it's just developed into one of the most important fields in modern biology. So I consider that is the accomplishment.
And your Nobel lecture, in fact, was, the title of it, as you well know, since you wrote it, was Nature's Gift to Science.
Yes.
And the first paragraph contains some typical Brenner witticisms and punning, which says, the title of my lecture is Nature's Gift to Science. It's not a lecture about one scientific journal paying respects to another, but how about the great diversity of the living world can both inspire and serve innovation in biological research. And you then go on to talk about the fact that, I suppose in the 60s, you hit on the idea of using Caenorhabditis elegans, C. elegans, the worm, as a model organism.
Yes, well, it wasn't just hitting on the idea like that. But it actually came about because Francis and I had started to talk about, now that everything had been solved, or so we thought in the early 60s, what would be the challenging things to do next. And in fact, there is a letter which I wrote to Max Perutz, which you can see, which said, this is what we want to do. In fact, it laid out the program for C. elegans, and of course we delivered everything we promised, even though some of it took nearly a quarter of a century to mature. But I think the idea of bringing to bear on an organism that was finite, simple, fitted the way we could work with it in a laboratory, and yet would allow you to probe all the things using, of course, the methods and approaches that had come with earlier molecular biology. In other words, you know, what does the DNA script mean, basically? That's what one is still interested in finding out. And I think that is still the unique, challenging problem.
In his remarks, the three of you made remarks there, Bob Horwitz said, quoted from Darwin, the man is but a worm, quote from Darwin. Here on behalf of the worm, John Sulston said. So there's this community of the worm runners.
Yes.
Has become an enormous part of science. I mean.
Yeah, I think so, and I think it was because Francis said Sidney likes to work with worms because he likes to watch them wiggle. And I think watching them wiggle, the actual direct contact, because now you are separated from the object you're trying to study, and you are separated by a vast set of instruments and computers and so on. But just to have the direct contact with nature itself, I think is what makes everybody go for it, because I think, you see, a novice could, and of course we were all novices when we started, but you could, with a few tools, very simple tools, you needed a good microscope to see them, but that was reasonable to get, and you could, with a few things inherited from working with phage and so on, you could start to do profound genetics within a few weeks. And you could find mutants and you could cross them and you could start. Now, of course, the journey beyond that, beyond the observations, that took quite a time to mature, and I think we were very lucky in many ways that it allowed us to get to the bottom of things quite fast, because simply of the whole development of the DNA technology and giving us the possibility of reading genes directly really changed us. In fact, it just made embryology a real science. And I think that was true for nearly all of our biology. And of course, we've now gone even further than that, because we can even study the most non-invaded species of all times, which is ourselves. And I think this opportunity to go to the gene script and try to find out what it means is, I think, still the problem that will take some time to solve.
In your Nobel lecture, you actually talked about two projects which were linked together. One was called CellMap and the other was called Humanity's Genes. That's still what's driving?
Well, CellMap is still, I think, something I would very much like to do. It, I think, is a framework for producing an integrated biology, integrated view. And I think in these days of information overload and the data collection, I think that it is still necessary. And the other project, which is related to this, is to stop talking about human genome, but to talk about humanity's genes. Because the way I look upon it now is that if you think there are 6 billion bases in both of your chromosomes, and there are 6 billion people or more than that on the Earth, this means somewhere all the mutations have already happened and are there. Now, of course, we have to find them, and of course many of them won't have much of an effect on the organism. But the fact is that the variability of humans is so extensive and so important for us as a means of studying them, that I think to think of humanity's genes is a much better idea than to think of, the human genome.
Let me ask you the two things that seem to be, I sometimes wonder if there's a contradiction here, because I hear you say some very funny things sometimes about the way in which science has gone.
Yeah.
You used phrases, I'm looking at back some notes I made from a lecture you gave, low input, high throughput, no output.
Yes.
You talk about, in your Nobel speech, there's a thing about we're conscious today that we're drowning in a sea of data and starving for knowledge.
Yeah.
And I get the sense that you think that what I might call the unit price of discovery has gone up astronomically. So when you started working, it didn't cost much to come up with some extraordinary discoveries, whereas now there's millions of dollars being invested in what seem to be fairly small increments of knowledge. Now, on the other hand, don't you need to have these enormous projects like the Genome Project and so on, doing the data collection? So could you
Yes, I think that that was justified. There were two ways of being able to read the sequence of, let's say, the human genome. By the time these discussions had started, let's say mid-1970s, when people had begun to think about this kind of technology, the largest organism that had been done was bacteriophage lambda. It was in fact seeing what Fred's sequence of bacteriophage lambda and how it could empower the analysis of an organism that set a lot of people on, that was called the Holy Grail at the time. I wrote a review of that and I called it the Lonesome Grail because I think it was a hard slog. And you could think of doing it either by factory methods, or you could think, let's wait for technology to develop. As you know now, you can sequence a human genome more or less in quite a short time and at a much lower cost, simply because technology has increased. And of course what has also come about now are very cheap, really very cheap compared to what it was then, computing resources. I mean, you can get the power on a bench top today that you needed, in fact, to launch all the satellites in the world. We just wasn't sufficient. We just didn't have that capacity to analyze data. Now, of course, what has become, I think, now a kind of imperative for many people is don't use, you don't have to think, you don't have to have any hypotheses, just go and make measurements and collect data. So it makes it more like a charting, and this a charting expedition. And in fact, at one stage I thought we had misnamed the entire subject, that it should have been called genonomy, which was of course what astronomy was a surveying of the heavens, because that's what we would do. I mean, astronomers are still charting the heavens. The better the instruments they have, the further out they can go, the better they can analyze. I mean, and just see what has happened with the Hubble telescope. Tremendous advance in our knowledge of the map of the heavens. So we could have made maps of this, and we could have done all of this, but the technology didn't exist. And therefore, we had to introduce more a style of management, had to replace what technology would have done in an automatic way. So I think that was fine, and but there is a feeling now that, oh, we can just go on doing the same thing. And after all, if you count a number of genomes, I don't know what the recorded number of species of animals is. I think it must be in the millions. I think insects may even be as much as that. Well, you know, we could sequence everything. So once you have the power to give atom by atom descriptions of everything, you say, well, let's go and do it. Let's get it. The problem about that is that that is not knowledge, okay? Because although everybody is saying more is better, it's actually the least which is best. That is, do the least and calculate or predict, or whatever you want to call it, the rest. Now, of course, you can't substitute then for what is really missing here, which is a theory about genomes. As you know, there were two ways of interpreting the stars. The one theory, which took a long time to develop, and you know, involved the development of relativistic physics and all the cosmology arguments. You could call it astrophysics. It is a branch of physics. And the other way is to get some idea that it is the stars that determine your future. And indeed, we have the analogy now in the genome. It is believed that we could study the genome and predict your future. We can predict future, you know, your destiny. In fact, I once said that although the astrologers, the horoscope, said you may meet a dark stranger, what we're telling people, you are going to become a dark stranger. Of course, it's only probabilistic, so we're only giving you a certain percentage of it. So it seems to me that there is that peculiarity of trying to understand this. I think if you say, well, why do the human genome? Well, there are technical reasons why it is the best one, which it's because it's the one that has been paradoxically the most slowly evolving. Okay, so you have to get a rather different idea now of what you mean by evolution, all right? We always think evolution is progressive, you know, always forever onwards and upwards. But it needn't be, and just consider the following. Suppose I'm given the genome of a human and the genome of a fish. I can compare them, and I can tell you what's different amongst them. But what you want to know is, when these two were the same, were they a lot of little humans running around at the time, which then sort of degenerated into this fish? Or there are a lot of fish at the time that became human? We cannot ascribe the sign to this until we know one other thing. And that is, of course, the fossil record, because clearly what the fossils show us is there were fish at the time. Therefore, we've got a direction for the process of evolution. But in fact, one of the things we're coming to terms with, a lot of people are coming to terms with, is a lot of genomes going downhill. They're minimalizing themselves, they're throwing away, or what they think, I mean, what the genome thinks is extraneous. So, the genomes, it is as though, you know, we'd come through the various phases of the history of art that started with the primitives, and then the classical, and then, you know, the Rococo, and now we're coming to what? The decadent, you see? Of course, the way to avoid decadence is to have the neoclassical revival, which is, I think, some of us think we must just get back to the job we know what to do, which is that we have to understand what all of these sequences mean. They mean this in terms of function, all right? And therefore, that is what we have to understand completely, I think. So as one of my current projects is called Reading the Human Genome, because I don't think anybody has read it. I'm the only one who's read right through it, from the left-hand end of chromosome 1 to the right-hand end of the Y chromosome. I can't remember what's in it there, but there's some very interesting things. The reason we can't assimilate all of that information in one go is that, of course, it's very detailed, contains lots of things. So, of course, we now have people that know about one gene or one little set of genes in great detail. That's fine. But the problem of putting all of that together is not yet available. Because I think that, and of course I think that's the most challenging intellectual question of all time. Can we reconstruct from the human genome, or any genome sequence, okay, the past? All right? There are very good conditions that you can define for how you can reconstruct the past. Can I go to the human genome and say, oh, there was an episode that happened at this time when they had very much fewer genes. And then there was one even before that, where they had half again, let us say. And can we make that connection? Because if it is true that there are residual features that are still lying there in our genomes, then I think we should be able to do that. It's a technical question as to what you would accept as evidence, but why I'm putting it this way is that you do not now take a genome and thread it into a machine and say, tell me what you find here, and then we do Bayesian statistics on, you know, how many times this is there. That's not interesting. That is like putting a book through a mill and doing the word counts in it. You need to have the integrated coherence of it. And because there is a lot of noise in the human genome, still people believe that everything's there for a purpose, but I don't believe everything's there for a purpose. If so, the purpose is not clear to me. I don't think you can deduce the future from what is there now, because that depends on so many other facts. But I think that you now find yourself actually making a hypothesis. If X, this idea, is correct, let us say, whatever it is, that this gene duplicated a long time ago, then this should be true for certain other things, then you go and test it. Because if you try to compute it ab initio, it'll be obscured. It'll be obscured by the noise that exists, which you'll have to cancel out in some way. So I think that there will be new ways of analyzing genomes, and I think and they're already appearing. I mean, this is being done for much smaller genomes, like looking at a lot of yeasts, for example, or a lot of different flies. And I think we have started to do this by looking at primates. Still don't understand, the difference between me and a chimpanzee. I mean, might be hard to discern, but nonetheless, there is a difference. And it's a difference that's composed of lots of different things.
So how does what you just said dovetail with two things? One would be the industry in personal genomics, where people are actually looking at a genome and deciding whether or not they think that there's possibility of some disease coming down the pike. And secondly, the work that Craig Venter has most recently been publishing with the synthetic organisms.
Well, you see, and I told this to Craig himself, you see, I think he's a counterfeiter. He's created some forgeries. He's taken other genomes and he's made copies of them and he's put them into organisms. He's not created anything new. He's just shown that he's just like the man who copied Vermeer's. Of course, he's actually copied the real thing. So these are forgeries, basically. Of course, he signed his name to them, so we know they're forgeries. I think what would have been better is had he created one in natural style, so we could recognize, oh yes, that is the style of those class of organisms. And look, he's created a new one in that style. Of course, maybe genomes don't have style, but I think to copy it, it's a technological feat, because it has to be done exactly, or else it won't work. That's the final test. But I think the synthetic biology is not yet a synthesis in that sense. In the idea that they are saying, which is to synthesize things out of some more simple components. It's just a copy of another organism which has been grafted in through this means.
This obviously was the President's, whatever it's called now, Commission on Ethics, I think, met to discuss that, because prompted...
So I gave evidence to this. That's why I know a lot about it.
Right. It put me in mind of, and also the things about stem cells, and so it put me in mind of Asilomar, going back to recombinant DNA and so on, and which you were also involved in.
Yes. The claims I think were not built up in this way. You see, one of the things we have learned since Asilomar had the thing, we're putting DNA from organisms that have had no natural way of sharing their DNA. In other words, we're crossing barriers, genetic barriers, and therefore, you should be careful. I mean, I always thought it would have been great to have combined duck and orange DNA. It might have saved cooks a lot of time.
Duck à l'orange.
Yes, but, so basically, but we now know that this business of barriers to genetic transfer, so-called horizontal transfer, is nothing like a sealed off. Genes have been moving around between organisms, some carried by viruses, some carried in other methods, and that this exchange has gone on considerably through the courses of evolution. So I think that one didn't have that in mind, but I think if he had shown you could actually synthesize a new organism, then I think this would have been the case. Now, in the time of Asilomar, I thought we should actually do a dangerous experiment. So I thought of one which is the most dangerous thing I could think of. Okay, which is to take ricin, which is this toxin in castor beans, and which was used, if you remember, by the Bulgarian KGB, scratched this person and killed a person in London some years ago. That was ricin. It's a few it's a very toxic material. To take that and put it into a phage and put that phage into a common bacterium. Okay, and I thought that would be a real one, and let's see, whether we could manage this, create this. So of course we then had all these levels of containment that went, up to P4, but we invented ones which were higher than this. In P5, you autoclaved the experimenter after the experiment, and in P6 you autoclaved him both before and after the experiment. But those, now, a few years ago, I was just reading about a toxin that is found in Shigella infections, This toxin is a close homologue of exactly ricin. It is found in a defective phage, which is called epsilon, which is a relative of our friend lambda, and both are in a bacterium called Shigella, which is a very close relative of E. coli. So nature has been far more effective engineer, which says that is, that making these bits and pieces and joining the DNA is probably quite a common occurrence, especially amongst microorganisms, and if it's to the advantage of the microorganism, it will be selected. So it's Darwinian selection, and it's man intervening in Darwinian selection that's the big problem, not the creation of new types, because I think that's all explored.
Let's circle back for a moment and figure out what this trajectory was. In the Singapore Medical Association news recently, there was a question asked of you where they said that you once gave a commencement address called "The Worst Medical Student of 1950," a personal memoir.
Yeah.
Why did you think you were the worst medical student in 1950?
Well, I was, yes. I gave it here in San Diego, by the way. I became a medical student because that was the only way I could get into higher education, into science. My local town council in the small town I grew up in South Africa had a bursary which sent people to medical school, allowed them to go to university. I was interested in science, in biological science, and of course I still think that the experience I gained doing medicine was effectively, the best biological training I could have ever had. And I did deviate from the medical course after 2 years for an additional 3 years to do science, because it was worked out that had I gone on, I would have been too young to qualify as a doctor. So I needed to take some time out. So it was a good excuse.
So you actually went to, this town is called Germiston, right?
Yeah.
Which is near Witwatersrand?
Yes, well, it's part of it.
Near Johannesburg.
Yeah.
And you went to the university at the age of 15? You were somewhat precocious, weren't you?
Yes.
And so by the time you'd finished this medical school, you were still too young to actually
I would have been under 21.
Right, so you then had to go and do something else. And
so I did something else which was convenient for me, of course.
And additional science courses.
And then, of course, at the end of this, this took me through till the war was over. And so there's a question now of, whether we should all go abroad and resume, studies in this. And so I thought that I then was told that given from the things I was interested in, which I called cell physiology then, that I needed a job in a medical school. And if you don't have a medical degree, you are a real 10th class citizen in a medical school. There were no PhDs in my university. So therefore I was advised to finish medicine, which I did sort of part-time. Okay, so when I came to I did fairly well. I mean, I wasn't the top student, but I was pretty close to it. And then, of course, I started to do clinical work. And I didn't take to that. I didn't like the funny kind of relationship of patients and a load of medical students, piling in to do this. So I didn't pay much attention to it. And in my final medicine examination, I was asked to smell the breath of a patient. It was a very famous story, which I did. And when he asked me, "What can you smell?" I correctly replied, "McLean's toothpaste." And the patient nodded in the bed because she'd cleaned her teeth before being examined. But of course, they just said I should have smelled, ketones, and she was a diabetic, you see. So having missed that, I had to go back and do it again, do another 6 months of it. But I did that.
Delivering babies, right?
I know I did. Did that very well. I went to deliver the babies, and that was something. There was nothing else to do, so I got very skilled at it, and I knew all about this. So that is why I think I was the worst medical student.
The interesting thing, going back even earlier, I mean, this is now December the 27th. So in a couple of weeks, it'll be your birthday. Again, January 13th. So you were born in 1927.
That's right.
And your father, as I understand it, this has been well documented in other interviews you've given, your father, Morris, was a cobbler.
Yeah.
And worked into his 80s. Apparently didn't think it made any sense to stop working at all.
That's right. Doesn't make any sense to stop working.
And he was from Lithuania originally.
He was from Lithuania.
Your mother from Latvia.
Yeah.
And he was not particularly religious, but she maintained a kosher home, I've read.
She did maintain it, yes.
And so your father was couldn't read and write.
That's right.
But he spoke several languages.
Yeah.
And you had a great love of books growing up, from, as I understand it, including a book called The Young Chemist by Sherwood Taylor.
Yeah.
And you got yourself a chemistry set. So chemistry, I'm just looking for what the initial impulse is here that sent you off in a science direction.
Well, chemistry was one of the things, but I used the chemistry I was very interested in why flowers had colors. And I thought I could extract them, which I did. And then I discovered I hadn't heard a litmus paper, but I discovered that they change color if you put acid into them. So I took this as a profound invention, but I was always doing things like that. I mean, using chemistry in the natural world, but I think chemistry was the sort of driving thing, because you can actually make something with it. And I think that was...
The Science of Life.
Yeah.
You told me this once, was a book that you actually stole.
I actually did steal it.
From the library.
Yes. it's one of the books well, shall we say I failed to return it and offered to pay. I mean, I've sometimes used this a lot of times, but my honesty did increase, and I thought, you know, I once wanted to get a book out of the library in Berkeley, the town library, and sort of even thinking that what shall I say? Shall I say I had the book and it blew out of the car while I was crossing the Bay Bridge, and there's no way I can retrieve it, you know? But I mean, I had cooked up stories like this, but this one I failed to return, yes. But it's a fantastic book, that.
This is by H.G. Wells?
It's Wells, H.G. Wells, and his son, G.P. Wells, and it's also Huxley. Julian Huxley, yeah.
There's a theme throughout all of these things that I read about you, about your passion for reading about things, the books, the papers. We've actually had this discussion about the way in which, although there are many good things to be said about the World Wide Web and the internet and so on, that I think your feeling is that people are not reading the papers adequately anymore.
Well, I'm old-fashioned. I like the feel of the book. And in fact, I think since I've come here, I'm reading about 5 books simultaneously, because I just, you know, you see amazing things in this. But I think the book threatens to disappear. This is what concerns me. And I don't know whether it actually will, but reading what people are doing about libraries and, you know, this whole idea of electronic, what do they call it, the digital library. Well, maybe the moving finger writes and then moves on. Maybe that's the digital library we should keep.
All right, so we've got you. You did your science year. Playing poker and doing skits with friends of yours, apparently. And then you get to Oxford in 52, August of 52. Yeah. In April of 1953, this is rather a significant time, you get into a car on a cold April day with Jack Dunitz, Leslie Orgel, who was another colleague at the Salk, and somebody else, Beryl Oaken.
We know who the Beryl, yes.
Oaken, yeah. And drive off to Cambridge, to the Cavendish.
To see the model.
To see the model. Could you reconstruct?
Yeah, well, of
DNA, of course.
Yeah. The thing is that when I came to Oxford, I had 2 completely wrong theories, but don't worry about them. But I was very interested in DNA and protein synthesis and how one might do the structure of them. And in fact, it was, I had all of these ideas, and I got to know Jack Dunitz because there was a place which still exists. It's called Halifax House. It was a place where scientists who didn't have a college or otherwise, and they were nearly all graduate students or postdocs, which Jack was. Jack was working with Dorothy Hodgkin. He was a crystallographer. Yes. And we discovered that, you know, we got talking and he did this, and then he introduced me to Leslie Orgel, who was at the Salk, and so we all got talking about this. So Jack knew, because he was in contact with the whole thing, because he'd been at Caltech, you see, that what was going on. And he then told me that somewhere towards the end of the year, I can't really date it, but they had apparently, you know, done something to solve the structure. But it must have been that we talked all through November, And so we then decided we must go and see the model. The model had been built, and all the things that you know had been. I'd never met Francis or Jim before, and so we went over there, and as I've written about this, this was an absolute revelation. I mean, I just said, this is it, you know, there's nothing more. And indeed, you know, when I used to go and give talks later in London, people used to say, what are you going to do? I said, I'm going to preach to the heathen. Because the strange thing is that nobody recognized that it seemed to be, the message was totally missed by lots of people. It was so amazing that for a few years still, nothing happened to this thing, except, of course, a few people began to work very seriously on the coding problem as it did transpire. And a few people, like myself, and because they'd now conceived of a way of testing some of this, which essentially was to compare the two sequences of proteins and DNA, except we couldn't compare DNA sequences then, so we were going to do it by genetics, which of course was Seymour Benzer's great sort of discovery, at least to me, and this said, well, we can now look at the DNA through the eyes of genetics. And so I think that that is how this whole early stuff of molecular genetics started. It was in an attempt to try to find out what the correspondence was between these two sequences. And I think this, after that came about, then I think that it then became clear that this was the thing that would carry the lever into the next stage. And I think if you look at that period, 53 to 56, very few things happened during that time. But I mean, it gathered pace so that in a relatively short time, if you think about it, by the middle of 1961, we knew about messenger RNA, we knew about proteins, we knew what the ribosomes were doing. And so roughly about that time, we said, enough, you know. As I think I put it in this note, we said that there would be hordes of American biochemists would enter the scene, and we'll go on and do something more interesting, which is how the C. elegans thing came about.
Yes, you like the opening game.
The opening game is a good game, because I think so few are allowed to play the end game, that in fact, because most of us are in the middle game. This was J.D. Burnell's idea of how to do science. He was very good at opening games as well, and just got bored by the middle game.